In this video, we will discuss about CD19-CAR-NK92 cytotoxicity mechanisms. In the previous video, we have understood about the two main mechanisms by which the CD19-CAR-NK92 works, and for that we have used the well-established cancer cell lines, and we are able to find out that the contribution by perforin and enzymes is much higher compared to the Fasl receptors. And however, when both of them are knocked down, then also they are able to see some cytotoxicity. So they wanted to target, or they in these results that are presented in these figures, we will see what approach they did to find out the cytotoxicity in the dual knockout, or the mechanisms that cause the cytotoxicity even in the dual knockout. So first, what they did is they validate their previous mechanisms, that their previous contributions of the mechanisms that they find out using the primary B cells. So in this case, they took the primary B cells from the patients and co-cultured them with the reference CAR-NK92, that is CD19-CAR-NK92 without any knockout, and this FSL knockout CAR-NK92, PRF Perforin knockout CAR-NK92, and the dual knockout CAR-NK92. What they are able to see in the wild type NK92, they are able to see a cell death of around 28%, and the reference CAR-NK92, this cell death increased to like 43%. But when the FSL is knocked down, it is decreased, and this decrease is statistically significant, and it is further decreased in the case where perforin knockdown is there. And when they compare the dual knockout with the perforin knockdown, the difference is not statistically significant. And when they check the contributions, they are able to find out the similar contributions, that perforin and Granzymes pathways plays a significant role. It is around 75% in this case, and 18% cytotoxicity is by the FSL ligand knockout, and around 7% is by the others. So, and this figure is the figure 4 of the main manuscript, that is figure on the left hand side, and figure on the right hand side is the figure 5 of the main manuscript. So from the from this human primary B cells, they confirm the contributions of the mechanisms, which is similar to what they have seen from the established cancer cell lines. Now they wanted to target this 7% other cytotoxicity mechanisms. So from the literatures, they come to a point that trail mechanisms also plays a role towards this cytotoxicity. So what it is, they use the antibodies that inhibit the trail on the Daudi cells and the CARtcells, and what it is, they co-culture this. For these studies, they use the established cell lines, that is Daudi cells and CARtcells, at different effector to target ratio 1:4 and 1:1. And in one case they use the trail inhibition antibodies, and in one case they don't use it, and they are able to see some decrease, but the decrease is not statistically significant for both Daudi cells and CARtcells when they use the reference CD19-CAR-NK92 that does not have any knockdown. However, when they use the dual knockout, it is FASL ligand and perforin 1 knockout in the CD19-CAR-NK92, they are able to see a decrease in the cytotoxicity. Although the decrease is subtle, but it is statistically significant for both effector to target ratios of 1:4 in the Daudi and effector to target ratio of 1:1 in the CARpus 422, indicating that this trail pathways also might also have a role towards this cell cytotoxicity by CD19-CAR-NK92 against the target cells that has the CD19 expressions. So these are the two ways by which they from the first figure from the figure 4, they confirm their cytotoxicity mechanisms or cytotoxicity contributions by different two major pathways, and the figure on the figure 5 is an attempt to find out what are these other cytotoxicity contribution pathways, out of which they are able to see the small subtle but significant contributions of this tail inhibitions pathways. So this is what they finally conclude in this paper about the different mechanisms, of which the major contribution is by Perforin Granzyme pathways, and then by Fasl ligand, and to some extent by the trail pathways also. So I hope you understood the results that are presented in this figure, and also the overall picture that is presented in this paper. If you need to understand more about these experiments in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.